Hello and good evening. I'm Dr. Naveed Akbar.
And I'm Dr. Nikita Ved.
Welcome to Pint of Science Oxford Live.
Thank you to everyone for joining us, and thank you to Warwick for the brilliant talks just now.
This Oxford event focuses on what makes us human. Is it our genes? Are we slaves to our body clocks? And do our emotions get the better of us? Today, we have 3 speakers who will touch on these areas.
We'd love to hear your views at home, and we'll be taking questions from you, the audience.
But first, we would love to know where you are in the world or where in the country you're joining us from. So please let us know in the comments section.
And please post throughout the talks on Facebook or YouTube. And Lucy, who is moderating the comments, will select a couple of questions for our speakers to answer. And if time allows, we'll select more than one question at the end of the presentations too. So keep your questions coming.
First this evening, we have Dr. Anna Machin, a doctor of evolutionary anthropology. Ordinarily, when we're having our in-person events in Oxford, Anna is our most popular speaker. So it's an absolute pleasure to have her with us today. Anna will discuss the darker side of love. When you're ready, Anna.
Brilliant. Thank you very much. And it's wonderful to be back at Pint of Science this year, even though it's a little bit strange circumstances. Those of you who've heard me talk before will know that I talk and research about love. That's my job, to research the answer to the question, what is love? But in the recent couple of years, I've become really interested in maybe the darker sides of love and how our need for love in our lives can lead to negative consequences for us. Ultimately, love is all about control. Evolution came up with love to reward us for starting and then maintaining those relationships in our life that are critical to our survival. And it does this at the very basic level through a set of neurochemicals, which, as I've said, reward us for starting and maintaining those relationships. At the very most basic level, love is biological bribery. And it bribes us to cooperate with those people who are most important to our survival. So with our partners, with our best friends, with our children, with our families. And we need to cooperate with each other to survive. We need to cooperate to raise our children. If anyone's ever cared for a newborn human baby, you'll know how incredibly dependent they are. And throughout our lives, our children need a lot of inputs, not only from families and friends and parents, but also from a whole range of professionals, including teachers and doctors, to make sure that they reach adulthood fit and ready to survive. We also need to cooperate to socially learn, to learn everything we need to know in this complex social and technological world. Now, in our closest relatives, the chimps, they do not take part in, for example, teaching or imitation. They learn the skills they need, such as nut cracking, from scratch on a trial and error basis. Now, if we tried to do that ourselves, it would not be a particularly survival beneficial thing to do. So we learn from each other. We also need to cooperate to subsist. Now, even today, when you sit on your sofa and you can get that grocery order to your door, as many of us have been doing during this time, if you think of all the people who are actually involved in that supply chain and supplying you, even though you haven't actually spoken or interacted with them in person, you are still cooperating with your fellow humans simply to subsist. And the fact that we require each other to survive, the fact that we require this cooperation underpinned by this set of neurochemistry that rewards us for doing this sometimes tricky thing, has led to the fact that the nature of our relationships is the biggest influence on our mental and physical health, our happiness and life satisfaction. This was first pointed out in a seminal meta-analysis, which was carried out by Julianne Holt-Lunstad in 2010 and her team. And what they looked at was 148 studies, which were looking at rates of mortality for people who suffered from a range of chronic and life-threatening conditions, such as cardiovascular disease, renal failure, and cancer. And what she found was that having a strong and supportive social network was as important, as impactful upon your health as, for example, quitting smoking. So it actually decreased your risk of mortality by 50%. And if we look at, for example, maintaining a healthy BMI, then having that strong set of relationships in your life where you reciprocate, where you cooperate, is actually more important than being of a healthy weight. Finally, I think one of the things that really shows how important love is to us in our lives is the concept of biobehavioral synchrony. Now, this was first developed by the Israeli neuroscientist Ruth Feldman, has probably arguably done more than anybody else for us to understand the neurochemistry of love. And what she's found was this, and I can imagine we all know this, that when you look at a couple who are closely bonded, who are very much in love with each other, whether it be a couple or a parent and child, we see they have behavioral synchrony. They tend to copy each other's gestures, adopt each other's tone of voice or choice of vocabulary. And this tends to bond them closer together. And what Ruth then did was she tried to understand whether there was actually physiological synchrony in closely bonded people when they are together. And what she found was indeed their blood pressure, their body temperature, and their heart rate tend to come in synchrony. Being a neuroscientist, obviously she cannot leave the brain alone for very long. And so she decided to look at whether brain activation and the neurochemicals that are released during those interactions also come into synchrony. Now, she looked in particular at oxytocin, and this was mainly because oxytocin is actually the easiest one for us to access. And what she found was that when couples come together, their baseline levels of oxytocin come into synchrony. Now, we all exist at different levels of baseline oxytocin, and that's one of the reasons why some of us, for example, are more sociable, or it might affect the way we feel love in our relationships. But these baseline levels come into synchrony in a closely bonded couple. We also see commonalities in brain activation, particularly in the gamma waves, which represent cognitive processing in the brain. So what this shows is that our love is so important to us, is so fundamental to your survival, that it infiltrates every fiber of your being. Now, this obviously shows how incredibly powerful love is and how incredibly important it is to our survival. But unfortunately, there is a downside to the fact that we require love in our lives. Because we have this craving, which is generally underpinned by the set of neurochemicals which are addictive and underpin love, it means that people can in fact use our need for love to manipulate, coerce, and abuse us. They can use love to control us. I want you just to read this. It says, early this year, 10 people located around the United States were arrested and charged in an organized money laundering scheme as they were attempting to wash the cash that they illegally obtained. What was strange about the scheme is how the money was obtained in the first place. It wasn't through the trading or trafficking of illegal goods or drugs, but rather cash that was sent by unsuspecting women who thought they were building relationships with the scammers. The victims sent more than $1.1 million, including one woman who spent more than $546,000 in cash and goods on a man who she thought she was dating. In 2019 alone, US scammers cost people within the United States $201 million, and that was 40% up on in 2018. And these people work by really playing on the yearning we have to be in relationships. So they will put up fake profiles and photos, they will slip into people's DMs, and they will start a relationship with them. They will pledge that enduring love, the fact that the people are absolute soulmates. Obviously, they will avoid meeting, but ultimately, after a period of time, and when they feel the moment is right, they will ask for money. So one of the questions I always ask people when I am exploring love with them is, can love ever be negative? Now, there are a group of people who actually answer no, and we'll deal with those a little bit later. But certainly people who say, yes, love can be negative often point to one of the first things, and that is the concept of jealousy. Now, I'm sure we all know that jealousy has evolved to make sure that we focus our attention upon our survival critical relationships. It is evolutionarily adaptive. And it's evolved to prompt us to carry out one of three behaviors: to cut off a rival, to make sure that we protect our relationship with our other half, or indeed to decide to cut our losses and go and find somebody else. And jealousy comes in three forms. There is emotional jealousy. So that is the fear, the anger, the sadness that comes with feeling that you have been betrayed. Secondly, there is cognitive jealousy, and that is things like contemplating what has been done to you, thinking about revenge, comparing your partner's new lover, for example, to yourself. And there is behavioral jealousy, and that is things like aggression, like monitoring behaviors, like feeling like you might confront the rival, for example. We also know in terms of jealousy, there is a strong gender difference in terms of how jealousy or when jealousy is experienced. Both men and women actually experience the same intensities of jealousy. There is no difference there. But where the difference occurs is in what circumstances jealous responses are brought about. And this has very much to do with whether the infidelity is sexual or emotional. And one of the leading explorers of this gender difference and the reasons behind it is David Buss, who those of us who have an anthropological background will know is one of the leaders in understanding human mating behavior and romantic relationships. And he's carried out several studies looking at what drives this gender difference. What is it that makes these differences arise? So, for example, in one unpublished study, he recruited 511 men and women and presented to them two scenarios. One was a sexual infidelity, one was an emotional infidelity. And he asked them to point out which was the most arousing in terms of jealousy for them. For 83% of women, it was the emotional infidelity that was the most arousing, compared to just 40% of men. In contrast, 60% of men were aroused in terms of jealousy by their sexual infidelity, compared to 17% of women. Oh, sorry, shot ahead there. And what triggers the physiological response differs. So we all get this physiological response when we are jealous. And again, this is to make us feel uncomfortable and make us prompted to actually protect the important relationship. So we might feel nausea, we have sleeplessness, we have headache, hot flushes and tremors. But again, what brings those physiological responses is dependent upon the type of infidelity, whether you are a man or a woman. So again, Buss recruited 60 men and 60 women and hooked them up to lots of different forms of machinery to measure their heart rate, their skin conductance, and the contraction of the frown muscles within the face. And again, what he found was this, that when we're talking about sexual infidelity, for men, this is the one that is the most physiologically arousing. So again, we saw an increase in heart rate of 5 beats per minute, which doesn't sound like much, but that's kind of the equivalent of you downing 3 cups of very strong coffee in close quarters. They found that their skin conductance went up by 1.5 microsiemens, which shows that they were sweating, and that their contraction of their facial muscles was 7 times more in the sexual infidelity scenario than in the emotional scenario. In contrast, for the women, it was the emotional scenario that drove the biggest physiological response. So the question arises, why do men and women have this gender difference? And we have to turn to evolution to understand that. And it depends upon what men and women bring differently to the reproductive relationship. Women, to put it bluntly, bring their womb. And therefore, if a woman is unfaithful, sexually unfaithful, she can deny her partner access to the womb. She will be carrying somebody else's baby for 9 months, if not longer. She will deny him the ability to transfer his genes down the generation. And for this reason, men find sexual infidelity the most arousing, because it affects directly their reproductive success. In contrast, a woman's reproductive success is controlled or affected by something very different, and that is the protection and resources that the male brings to the relationship. So for a woman, it is emotional infidelity that is most arousing when she looks at her male partner. Because, okay, he may have sex with somebody else, but that's not going to actually directly affect her reproductive success. However, if he builds a deep emotional bond with somebody else, then he puts forward the prospect that she will have to share the resources that he brings to that relationship, which will directly impact negatively on her reproductive success. But for some people, jealousy becomes out of control, and it can lead ultimately to abusive, coercive behaviors. And the question arises, are some people more likely to be abusive in relationships than other people? And this brings us to the concept of the dark triad. Now, the dark triad explains a group of personality characteristics which, when carried all together, can increase the likelihood of somebody using costly mate retention behaviors upon their partner. And the dark triad is 3 very well-known personality types. It is Machiavellianism, psychopathy, and narcissism. Now, I'm sure we all know Machiavellianism is having no empathy for somebody, but actually having incredibly strong emotional intelligence and using that to manipulate. Psychopathy is, again, having no empathy, but also having quite antisocial behaviors, but really enjoying sporting with people's feelings and manipulating them to your own ends. Somebody who is a narcissist is greatly self-aggrandizing. They have huge self-regard, and they tend to denigrate other people's self-regard in comparison. But what the question arises is, do people who carry all three of these, are they more likely to carry out these costly mate retention behaviours? So we can split mate retention behaviours into costly and beneficial to the partner. So a beneficial mate retention behaviour, for example, is empathising with them, is caring for their day, is supporting them when they're ill, is giving them gifts. A cost-inflicting mate retention behaviour is something like surveillance, control, physical, mental, psychological abuse. It can be denying access to possible potential other partners. It can be controlling what somebody wears, or maybe controlling where they go. And these are cost-inflicting behaviours upon the other party. And in fact, in a study in Iran in 2018 by a group of psychologists, they recruited 205 men and women to understand whether possessing all three of these dark triad traits did increase the likelihood that they used cost-inflicting mate retention behaviours. First of all, what they did was they gave them a wonderfully sounding set of questions known as the Dirty Dozen, which diagnoses whether people carry the dark triad personality traits. They then asked them a series of 38 questions, which were split into 19 different sorts of mate retention behaviours. So, for example, the positive ones were things like gift giving. But if we look at some of the negative ones, they were things like, I snooped through my partner's personal belongings. I slapped a man or a woman at a party who was flirting with my other half. I flirted with another man or a woman at a party to make my partner jealous. And what they found was that people who carried the three personality traits of the dark triad were much more likely to use these cost-inflicting mate retention behaviours. This wasn't to say, however, that they never relied on a beneficial behaviour. Indeed, their rate of beneficial behaviour, whilst much lower, was still there. And in fact, we see this in abusive relationships. For a lot of the time, the partner will use cost-inflicting mate retention behaviours, but they will scatter beneficial behaviours through the mix, because this destabilises the person who they are in the relationship with and can lead, for example, to the phenomenon of gaslighting. The interesting thing to say is that because these people have persisted within our population and within our species, suggests that to them, while not their partner, having the dark triad set of personality traits is in fact a positive thing for your reproductive success. I want to introduce you to some statistics. The WHO carried out a study of 24,000 women from 10 different countries around the world, from Bangladesh to Ethiopia, Serbia to Japan. And they came up with these statistics when asking them about the incidences of abuse within their lives from their partners. 61% had been physically abused and 75% of emotionally abused. 3/4 of the women who were questioned had experienced some form of emotional, coercive, abusive behaviour. In a study carried out by the US Centre for Disease Control and Prevention, looking at men and women, they found that 1 in 5 women and 1 in 7 men have experienced physical violent abuse within a relationship. They defined that sort of abuse as being punched, slammed, beaten, kicked, burnt, choked, and attacked with a weapon. And when we look at emotional abuse, so that's psychological abuse, coercive control, both men and women reported that 48% of them had experienced that within a relationship. When I ask people whether love can be negative, sometimes they say no. And when I bring up the prospect of, for example, people who are in abusive relationships, they will say, well, that cannot be love. But the thing is, love is a highly subjective thing. Whilst we as scientists have worked very hard to understand the underpinnings in terms of genetics and neurochemistry and neuroscience, in terms of what actually controls someone's experience of love, there is a major element of your love experience which is entirely subjective and that we will never be able to measure. How I feel love and how you feel love are probably entirely different. And because of this, it is not for us to speak to somebody else's experience of love. And for me, as somebody who researches love and is now interested in the darker sides of love, it really does interest me where love sits in these abusive relationships. What I'm particularly interested in is looking at male abuse victims. And as you can see here, this is Colin, someone who I've recently been interviewing, who is a survivor of domestic abuse. And in a 2013 study of women who had been in abusive relationships, a team of 3 mental health nurses from West Virginia University talked to 19 abused women to try and understand their concept of love within relationships where they experience intimate partner violence, or IPV. Within their study, they defined IPV, as you can see on the screen there. So slapping, intimidation, shaming, forced intercourse, isolation, monitoring behaviours, restricting access to healthcare, opposing or interfering with school or employment, and making decisions concerning contraception, pregnancy, and elective abortion. They asked the women 3 sets of questions. First of all, they asked them to describe for them what they thought love was. And they explained it in the way that many people explain it to me when I ask them to define love for me. So it's about respect and understanding. It's about support and encouragement. It's about commitment, loyalty, and trust. They then asked the women to explain what love wasn't. And they, many of them speaking from experience to a certain extent, said it's being hurt and scared. It's being controlled. It's being hit. It's being not trusted. And it's someone who shows a lack of concern for your welfare. Having asked the women to compare and contrast these 2 definitions of love, they then asked the women to explain to them where love sat in their relationships. And as you can see from this particular quote from the study, love is something that these women were feeling as far as they were concerned. The question arises, why do people who are abused stick into these relationships? When we are talking about the women in this particular study, it was partly a need to protect and nurture their partner and be nurtured in return. For some, as the woman who's speaking here, it's a hope that their partner can improve, can get over the abusive side of their personality, and that they really feel that they are the person to help them to do that. Some of them, it's a fear of being alone and the very real belief that if they left this relationship, they would simply step into another that was even worse. It's in a way that it's better to be with the devil that you know. And many of them had very strong ideas about the importance of nuclear family. And therefore, in fact, their love for their children was one of the things that kept them in their relationships. And as I've said, we need to remember that men are victims of IPV as well. And certainly in my studies with survivors of abuse, male survivors of abuse, many of them have spoken in the same way about their need to be there, their need to maintain that belief in the nuclear family, the belief that in the beginning, love is blind. And we actually do have some neuroscientific evidence to back that up. Certainly when you are first in love, those areas of your brain linked to mentalizing, and that's the ability to tell what somebody is really like, do shut down. So there's certainly an element of that in play. But also many of the men spoke about the fact that the women used their love for their children to keep them in a relationship because the men did not want to leave the child with the abusive partner. And unfortunately, in many countries, it is still the case that children overwhelmingly remain with their partners. Another study has asked the question, is romantic love itself to blame for people who stay in abusive relationships? I've spent many years studying romantic love, not only the neurological underpinnings of it and the genetic underpinnings on it, but also what we as a culture believe romantic love to be. And particularly in the West, it's something that we are particularly obsessed with. And we believe that's the only form of true love. But the question arises, is it the narrative of romantic love that partly makes people who are in these abusive relationships remain? And one of the most powerful studies I've seen carried out by Singh et al. And published in 2017 was one that was carried out in South Africa amongst female undergraduate students. Now, South Africa has one of the highest incidences of IPV in the world, and being murdered by your partner is in fact one of the main reasons for female death within the country. And they were interested in talking to these women about building their resilience to IPV as they started on their journey through life. But one of the bits that really interested me in the study related to their cultural ideas about love, and how those cultural ideas about love meant that maybe they would stick with an abusive partner when it was better to leave. So, for example, if we think about our ideas about romantic love states that our romantic love will overcome all obstacles, that we will be able to get over all hurdles as long as we are with the person we love. And that might include getting over the problems of, for example, an abusive partner. We have the idea that we have no control over who we fall in love with. There is romantic fate, and we are soulmates, and we will be together, and fate will bring us together. Again, that we cannot control the fact that we love this person, even though they are not good for us. That lovers protect and fight each other to the end, even against the opposition, for example, of family, of friends, of the culture, and also possibly in the face of people saying that it is not a healthy relationship to be in. And of course, the idea that love is blind, and that we can turn a blind eye to maybe the negative parts of our partner's characteristics. Finally, just before I finish, I want to finish on a note really more about the future and our need to control love. Humans find it very, very difficult to deal with things that they cannot control. One of the reasons why we like science and scientific discovery is it gives us an explanation for the things we see or experience happening in our lives. And for over 2,000 years, we have sought an elixir of love, something that we can take where it will either make somebody fall in love with us, make us fall in love with them, or indeed blot out or stop the powerful feelings of love we might have for somebody when it is unrequited. And certainly there are many, indeed, plays and operas and bits of art that have been based upon that idea. But the thing is, today, as we have a bigger understanding of the neurochemistry of love than we ever have before, the idea of a love drug actually is tantalizingly close. And I'm sure you can imagine that many people, but also many drug companies, are interested in understanding how these drugs might work. The key players in this particular drugs search are oxytocin, MDMA, which is ecstasy, and the SSRIs, which are used to treat depression. So if we look at oxytocin, oxytocin is incredibly important, particularly at the start of a relationship, because it lowers your inhibitions to starting new relationships. It gives you a confidence based upon its quietening of the amygdala. And certainly those of us who've worked with oxytocin in studies looking at how oxytocin lubricates the wheel of social interaction say that in many cases, squirting somebody up the nose, for example, with oxytocin does increase their sociability and their trust. If we look at MDMA, then certainly ethnographic reports of people who regularly take ecstasy say that their experiences are full of powerful feelings of love and empathy for their fellow takers. And we know that people who take MDMA on a regular basis can have permanent increases in their empathetic abilities. And if we look at the SSRIs, then again, one of the side effects of SSRIs is that they lower libido, but they also lower the intensity of emotion in a lot of users. And therefore, SSRIs have been suggested as being a possible anti-love drug to be taken by people who maybe are finding it hard to get over a difficult breakup, or indeed wish to stop loving somebody who, for example, is not healthy for them. The issue with all of this is that we don't all react to these particular drugs in the same way. So, for example, in terms of oxytocin, there is an increasing number of studies now showing that oxytocin actually, in some people, increases ethnocentrism. Therefore, it makes us more aware of our in-group, out-group differences, and therefore less likely, in fact, to trust others and be open to social contact. MDMA only increases empathy and sociability in a certain group of individuals who carry a particular SNP of the oxytocin receptor gene. Those who do not carry this gene, which is also this SNP, which is also implicated in a whole range of pro-social behaviors, actually it has no impact at all. So what we need to say about these drugs is their impact upon us are highly individual and context dependent. Certainly David Nutt, who, for example, has looked at the role for MDMA in pro-sociality, has said that it's very difficult to get those empathetic results in the lab setting. And I also want to tell you a little story about the use of ecstasy, just to show the possible ethical issues that are involved in using love drugs. So there was a guy and he liked clubbing, and every weekend he took ecstasy. And one weekend he met a girl at the club and he fell in love. And for the next 2 years, they had a relationship where they met up every weekend. They lived in different cities and had the most wonderful time in a very deep and loving relationship. They were boyfriend and girlfriend. Then towards the end of the 2 years, they decided they should take this further and move in together. So they did. And it became quite quickly apparent to the man involved in it that he did not love the girl. Because the issue was he did not take ecstasy or MDMA during the week. Now, we can quite say that we can use whatever we want to put into our own bodies. And yes, we have freedom of choice. But the issue here is that that choice to take that drug, it wasn't something he did consciously and obviously regrets it. But if we consciously take a drug that alters how we feel about love, we need to understand that impacts on somebody else's life. His girlfriend had changed her entire life to be with him. And it was actually based upon a false premise. So what I want to say is we need to have big conversations about using love drugs as they come onto our horizon. It's probably an ethical conversation we have to have now rather than later. For example, SSRIs, whilst they may be great for stopping us loving people who we might not want to love, can also be used by repressive regimes who maybe want to stop, for example, homosexual love being something within their society. There are still 72 countries in the world where homosexuality is illegal. And certainly there are reports from some particularly strong faith groups where SSRIs are being used to suppress homosexual feelings, particularly in young men. We also have to be worried about the power imbalance that might occur if one person takes a drug and the other one doesn't. And where do we draw the line? If, for example, we decided to use MDMA in therapy, for example, there are some proponents saying that this would be a good move in terms of helping people be more empathetic in therapy. And it certainly has good results. But where do we draw the line? Is it going to be difficult to close that door once the horse is bolted? And are we happy, for example, to have that use within parent-child therapy? So the question I want to leave you with is, would you take a love drug? That was a real rattle through the dark side of love and control. If you are interested in any aspects of control and love and the use of drugs, then please go to my website, which is there. There's a 16-part YouTube series on there, which was filmed in conjunction with Pint of Science. One of those episodes is about love drugs. And if you go to my website, there's lots more information on my work on love. Thank you very much.
Thank you, Anna. Absolute brilliant talk as always. Really, really enjoyed it.
Thank you.
Absolutely spoiled for questions. So thank you to everyone. We have over 300 people with us and we've had over 50 comments. But I will pick one here from Alex Holmes. Are the jealousy responses different in same-sex relationships? And if you extend that, what about individuals who are non-binary?
Okay, I would say, and this is always a question I get when I'm talking about love, unfortunately, the research that's been carried out on, particularly, for example, gay relationships isn't particularly strong. If I go from other knowledge, for example, about mate choice behaviors in romantic relationships amongst gay people, actually, they tend to react along sex lines. So you will still get this sexual infidelity feeling. So, for example, if you've got a male gay couple, both will still find sexual infidelity harder than emotional infidelity. And that might be because of something to do with a deep-seated evolutionary story behind that. Unfortunately, research into homosexual love is still in its early stages, which is obviously a massive source of regret and something we need to speed up. In terms of non-binary, to be honest, I don't think it's been researched yet. One of the things I would say about non-binary is very much talking about a gender. And obviously, we have to get into that discussion about gender and biological sex. Again, I don't know what the reaction to that would be, whether somebody is culturally identifying with gender, which is what gender is. It's a cultural thing. But obviously, their sex might still dictate which one of those they find more powerful. I know that's not a very helpful answer to your question, but we're in the very early stages of understanding, particularly non-binary behaviors.
No, I think it's very important to ask all types of questions. So this type of jealousy, this form of love requires a better understanding.
Absolutely. Yeah. And we're still, unfortunately, what always happens, as all scientists who are listening to this will know, you start with generally your biggest group, which is heterosexuality, and we work our way down through that. And we need to focus a lot more on it.
Thank you. So we'll take one more question just quickly from you. This one's from Vicky Mools. So thank you for your question. Is there any research on the differences between self-love and love from another person in relation to the benefits that you've described?
Again, I don't think they have. I mean, I think I would probably answer that in terms of self-love. We use the word love in lots of contexts, and it's one of the questions of what love is, because we tend to bandy it around a lot. If we look at love in the scientific sense, self-love isn't love in the scientific sense. Self-love tends to be a psychological state where you care for yourself, which is great, but it's not going to generate the underpinning of neurochemistry, which love to another person or a relationship with another person does. So while self-love is probably incredibly good for your mental health, it's incredibly good for you psychologically, for stress and things like that, in terms of the actual survival benefits that come from being in love with somebody else, whether that be a partner, a child, a parent, whoever it might be, you're probably not going to see the same thing, because love for another gives you major benefits in terms of that biobehavioral synchrony. You get all the wonderful floods of neurochemistry, which, again, particularly underpin your immune system. So, no, the sort of benefits you're going to get from self-love, while wonderful, are not going to be as powerful as being in love with somebody else.
Well, thank you very much again, Anna.
Thank you.
Hi, everyone. So next up, we have David Ray, a professor of endocrinology. And David will talk about the rise of body clocks in evolution and how they've proved essential for life on Earth, and how modern day shift patterning work has influenced that now. So please take it away, David.
Thanks very much indeed. I was going to attempt to follow that up with a short talk really relating to how time affects us in biology and also in medicine. Okay, so the observant among you will have realized that what we have here is a predictable change in the environment between the day and the night caused by the rotation of the planet on which we live. And it turns out that actually all throughout evolution, we've evolved a means to anticipate this change. So in humans, we have a clock ticking in our brain here. And that's why if you're asked the time, in most cases, you can get the guess right within an hour or so. So what does this clock in our brain drive? Well, this is a 24-hour clock here. So this is the day over this side, and this is the night over here. So our ability to do particular things and particular physiological parameters like body temperature, blood pressure, and so on change throughout the day. Now, how does this work? Well, inside every cell in our bodies, we have our own molecular clock, and this is a cartoon of it here. The discovery of this molecular clock led to the award of the Nobel Prize in 2017. It's really just like a pendulum clock in that you have a feed-forward loop, like the pendulum dropping under the influence of gravity, followed by a feedback loop here, which is like the pendulum slowing as it starts to swing up the other side of the median. So your cellular clock actually has two feedback loops, one on this side, one on this side. Okay. So what about time, as in the time on the clock on the wall versus our biological time? Until very recently, clocks on the wall had, a very flexible relationship with time. This is a diagram of city time zones across the continental United States of America in the mid-19th century. So around the edge, these sort of pink circles, these are the local time zones operating in individual cities. And I've just zoomed in here. You can't see the exact numbers, but you can readily see that the time zones differ by sort of 15, 20 minutes, half an hour, and so on. So there was a huge number of different time zones in operation. And it was only the onset of rapid travel with trains that required harmonization. And in 1883, 4 time zones across the continental US were instituted. So that's fine, but this is what's happening in our cells. As I showed you earlier, we still have a molecular clock that ticks in our cells, and these are just some human cells, and these are just cultured in a plastic dish in my lab, and you can see that they're ticking on oblivious. So no one's told them about the clock on the wall. And of course, it's this fight between our internal clock and the clock on the wall that leads to trouble when you, for example, fly to the US and you get jet lag, or if you work a night shift and you can't sleep during the day. So it turns out that actually there's a whole load of clocks in our bodies. And the central clock here in the brain drives a synchronized oscillation in clocks throughout the body. And that results in clocks operating in every organ in our body, ticking in synchrony. Okay, so what do I mean about synchrony? Let's just have a look and see if Daniel can help us out. Try and get this to work. It's not going to work, is it? Oh, maybe it is. And here's Daniel showing us how this works. Okay, so these are metronomes. They're ticking away, and each of these metronomes is like one cell in our bodies. And you can see that they're all ticking out of phase, and it's making a terrible noise. What happens is when they're connected in some way, as if, in this case, by balancing on Coke cans, but in our body, it would be a shared circulatory system, and that allows all the cells, or all the metronomes, to tick in perfect synchrony. So that's what we refer to as internal synchrony, and that seems essential for maintenance of human health. Okay. Try and move on now. Can't get out of this. Okay, good. So, as I said, my background is in medicine, so why do I care about this? Well, it turns out that almost all human diseases are circadian. This is just an example looking at people with rheumatoid arthritis. And with people with rheumatoid arthritis, Their symptoms vary during the day. So they have a peak onset of pain and swelling in the joints here in the early hours of the morning. And that's accompanied by a peak here in the red line of the activity of the immune system that drives that inflammation in the joint. So if we switch our attention to the lung, a common lung inflammatory disease would be asthma. And again, if anyone in the audience has had asthma or knows anyone with asthma, they know that the symptoms are often worse overnight, causing nighttime coughing and sleep disturbance. And if we look at what happens in the lung, it's a very complex organ. But if you look at individual cells, these are cells lining one of the airways, and these are the sort of immune surveillance cells. When we put them in a dish, they oscillate. They've got their own clocks. And this is an example of a very important protein which regulates the immune response. And you can see it's either present or absent, depending on the time of day at which you look. And this is an extension of that observation to really look at how the lung responds to inflammation. And here, these are professional immune cells which come into the lung to fight off infection. And you see you got a lot more immune cells, and this is at dawn, compared to other times of the day. This is just enumerated here. And these cells come from the blood into the lung under the influence of these chemical messengers. And you can see a number of them are just plotted out here through circadian time. And you can see that the expression of these mediators changes a lot through time. So therefore, it's perhaps unsurprising that many drugs we commonly use in the clinic to treat people with inflammatory diseases, and this is just a summary of 7 trials here, show a marked time of day variation in how well they work. And now we've got some insights into why. This is a very interesting story involving a group of hormones or drugs now called glucocorticoids. So the famous one is dexamethasone, which has recently been used successfully to treat people with COVID-19. And when we use this, you can see that it inhibits, when we go from the black line to the gray line, inhibits some of the inflammatory mediators that drive the recruitment of inflammatory cells into the lung. And if we disturb the operation of the clock, you can see that now the dexamethasone just doesn't work. It's selective, so it doesn't work on all mediators. For example, another mediator here, that responds to the dexamethasone irrespective of interference with the clock. But critically, this is counting those professional immune cells flooding into the lung. See, we can block that with dexamethasone under normal conditions, but if we interfere with the clock, that effect is completely lost. So this remarkably important drug, dexamethasone, used to treat COVID, requires the clock to work. So we're all aware that we have a preference for either being up in the morning or active more late at night. And this here is just some data on a large number of human subjects. This is age across the bottom. And this is a measure of the time of day at which people prefer to be active. So the higher up here, then the later the chronotype. So as you can see, late teens, early 20s, these individuals like to go to bed late at night and get up late in the morning. The boys are in blue and the girls are in pink. So teenage boys are extremely late, which of course leads to delinquency because they fail to make school start times. So this has practical implications. I'll leave you to speculate why it might be that having adolescent boys and girls active at the time their parents are asleep might have some interesting social consequences. And this is just some very old data, but just to point out that it's not all about timing with a period of 24 hours. Now here we're looking along the top at months through a calendar year, and this is a sample of males, and they were asked to score the frequency of sexual intercourse by month. And as you can see, there's a very interesting circannual variation with a peak that topically is about to arrive in October. So just to summarize and conclude, circadian clocks are prevalent throughout all of life on Earth, everything from bacteria to plants and animals, and likely they've evolved to anticipate changes in the light-dark cycle. Our clocks govern our lives. Virtually all aspects of our lives are profoundly influenced by underlying biological timing mechanisms. In nature and throughout human evolution, sunlight was the key to keeping our clocks synchronized to the daytime. But of course, now we have 1 in 5 workers working night shifts, we have electric lights, we have exciting TV programs late at night, and therefore we have a struggle between modern technology and modern working practices and these billions of years of evolutionary selection. So it's no surprise that we're struggling. Perhaps one thing you may want to do after this talk is to look at your smartphone, go to settings, go to display, and activate the night shift mode. This will reduce the light intensity on the screen, so allow you to scroll later into the evening without disturbing your sleep. I'd like to thank very much colleagues in my group in Manchester and now subsequently in Oxford, who generated the work I've showed you, and to very generous funding from the Wellcome Trust and the Medical Research Council. Thank you very much indeed for your attention.
Thank you, David, for an absolutely smashing talk. We've got a lot of questions to get through, but I'll have to whittle it down. So we had our first question from Tessa Flack. So how would this affect people who have jobs that involve changing time zone a lot, e.g. flight attendants and pilots? Is changing time zone so bad for their bodies?
Yes. So the short answer is yes. We're right in the middle of this exciting experiment where we're expecting people to work and live against their clocks. And there's an abundance of evidence that this is harmful both for mental health and also for physical health. So as an example, there's now very clear evidence that night shift work exposure is an independent risk factor for the development of breast cancer in women, and a number of other malignancies, including breast cancer, have also been associated. We've just done some work looking at night shift as an independent risk factor for developing asthma. So yes, individuals who are either working nights or are exposed to frequent jet lag are at much increased risk of both mental and physical harm.
Oh, very interesting. It seems that these lifestyles may not be as glamorous as they may be perceived.
Well, that's right. And one of the problems we have at the moment is there's a real lack of understanding of how drastically our lifestyles have changed, and we don't fully understand how to mitigate the risk. We accept that hospitals need to be open at night, so therefore we do need to have some people working at night. But how do we look after the health and mental well-being of those people who are working nights? And that's something I'm passionately interested in, because I think it's a real critical lack that we really don't understand what to do for the best.
Oh, very good. I think we have time for one more question. And this is from Anne van Dongen. So why do people in general worry more before bedtime? Is this any relationship to serotonin?
I don't know. It's a really interesting question. So we spend a third of our lives asleep. It's absolutely essential as drinking and eating. And yet we really don't understand a great deal about what goes on during sleep. So what you're talking about isn't exactly during sleep, but in the sort of run up to sleep. And what I would say is that among the things that affect sleeping behavior is it can be a sort of a learned behavioral response, particularly in the preparation. The sort of neurohormonal changes in the brain start about 2 hours before sleep, with a rise in melatonin in the brain, which seems important for preparing the brain for sleep. So it's possible that it's some of the neurochemistry changing there. And of course, that's why the advice is to make sure you're in very dim light and you're avoiding screens, maybe some quiet reading. These kinds of things are the ideal ways to get in harmony with your brain's preparation for sleep.
Oh, that's very interesting. Well, thank you very much, David, for your time. And please do stick around at the end. If we have time, we'll ask more questions from the audience.
Sure, thank you.
Thank you. And last, but by no means least, we have Charlotte Rich-Griffin, a computational biologist. So Charlotte will discuss how gene sequencing technologies are enabling scientists to study single cells and how from this data we can build models of whole organs and tissues. So please, Charlotte, when you're ready.
Thank you very much. for the introduction. I am glad to be here giving my first Pint of Science talk, and we're going to take a slightly more light-hearted walk through the world of cells. I'm going to start by introducing you to Emmett. Emmett, in fact, is not very well. He has digestive problems, he's been feeling sick, and he's generally been feeling pretty terrible. And so he's headed off to his doctor. And after a thorough exam and a history, she decides that the best thing for him is to go for a colonoscopy, where they put a camera up into the intestines, and takes a closer look at what's going on inside. And what's more than that, they're going to do a biopsy, which is to take a small sample from inside, and that sample goes off to the lab. And here, highly trained pathologists will be able to take a look at that sample under the microscope. And they'd be able to tell you from this that Emmett is suffering from ulcerative colitis. And it's a pretty bad disease. It's chronic. It currently doesn't have a cure, and it will last for the rest of his life. But ulcerative colitis varies from person to person, and the differences between this is in your genes. You inherit different genes from each of your parents, and that combination of genes makes you uniquely you, and that can make the difference between developing a chronic illness or not, or responding to treatment or not. So let's head back to that biopsy. We could see from the biopsy that Emmett was suffering from ulcerative colitis, but we don't know what's going on inside the cells. And without looking at those cells at a genetic level, we have no hope of understanding his disease. So let's talk about cells. You have 37 trillion cells in your body. To give you an idea of how many that is, if every single one of your cells was a Lego brick, you would be about a mile tall. Picture the Eiffel Tower. You would be this big. And you have lots of different types of cells in your body, making you. You have the cells. Cells are what give you structure. And they take nutrients and turn it into energy. And you have specialized cells in your body that do things like the cells in your eyes, which detect light, and the nerves that take your light from your eyes to your brain, and your brain cells that tell you what you're seeing. In this case, a very tall Lego person. And what makes the difference? Because in your DNA, you have a complete set of DNA in every single cell. So why can't you see with your feet, or touch, or taste with your fingers? I'd like you to imagine a wall of light switches. Each one of these light switches represents a gene, and some are turned on and some are turned off. And this means that this is what will give an individual cell its special function. Every single cell has all of the light switches, but different ones are turned on and off. So let's return to that biopsy. In Emmett's case, his cells have started attacking himself, and they are all malfunctioning within his system. So wouldn't it be great if we could look at this at a genetic level, at a single cell level? Well, that is exactly what we can do. I work in a field that is dedicated to looking at cells at this resolution. And the way it works is this. You take a sample from the biopsy, you get a little blob of tissue like this, and you separate it out into the individual cells, and then you dilute them into a flask. And you can take those diluted cells and pass them through a machine where they get captured in droplets. And this is more clear in a visualization. Here you can see droplets are being made, very tiny droplets, and then cells come along the top, that's that black circle coming along the top, and gets captured in that cell. And the really cool thing about this is inside each of those droplets, there is a barcode. that tells you which cell is which, in the same way that a barcode will tell you which product you've got at the supermarket. And then it's a simple matter of working out what cells you had using your unique barcodes and your combinations of genes that are on and off. And that is a bit like turning this into this. And this might be able to give you some insights into what is going on in your biopsy. Maybe they've got too many blue cells, or maybe those dark green cells aren't supposed to be there at all and they're supposed to look like the light green ones. So this can make the difference between being able to identify 5 or 6 cell types under a microscope to being able to identify 30 or more. And you can see from this data how they are working together to make a healthy gut or malfunctioning in an inflamed gut. And this has the potential to inform new drug development and therapy development and personalized diagnosis. So let's take a step back and think about, what if we could do this for an entire human? You have a lot of different types of cells. How many do you think make up an entire human? 1,000 cell types? 10,000 cell types? 1,000,000 cell types? The actual answer is we don't really know, because since we've started looking at cells at this resolution, at the genetic level, we keep discovering new cell types. So let's think about our idea to try and do single cell analysis on an entire human, 37 trillion cells. Well, firstly, you've got to find a person who's willing to be dissolved, which might be a bit tricky. And secondly, even if you could do that, well, it's far too expensive to run that 37 trillion cell experiment. It would cost trillions and trillions. But we could be a bit more systematic about it. We can take some samples from different bits of the body, from the lungs and from the brain. And then we could do it in lots of different people at different ages. And we could start to build up a picture of not just a human, but a human across their entire life. And that's exactly what the Human Cell Atlas aims to do. The Human Cell Atlas aims to create comprehensive reference maps of all human cells as a basis for both understanding human health and diagnosing, monitoring, and treating disease. So let's go back to Emmett in the clinic, waiting for his results. What if we could take the information locked in his cells, into his genetics, to decide what would be the best treatment for him? What if we could cure him because we know exactly what genetics are driving his illness? And I think we could send him away pretty happy. Thank you very much.
Thank you, Charlotte, for your brilliant talk. So I think we have time for one question, and that is from, just bear with me 1 second, and that's from Alex Holmes. So how do you define one cell type as different from another? For example, how would you differentiate between an eye cell or a foot cell, and how do you define those things?
That's a really excellent question, and actually gets to the crux of what my entire job is. You can look at it on different levels. We could look at it on the level of the genes or on the proteins. And so you would say, I've got this set of genes that are turned on or off, or I've got these set of proteins that are turned on or off. And I know that in order to become a photoreceptor, which is one of the eye cells, you need these proteins. And alternatively, a stomach cell. And when you're looking at all of the data together, you use algorithms from like data science to try and cluster together based on whether they look similar or whether they look different. And looking at these clusters of cells, that is what tells us this is a cell type, which might be a cell type that we've never seen before. So genetics and proteins is the main way we would decide. Also what it looks like.
Wonderful. Okay, so I think that might be it for questions. I think we'll...
Thank you for joining us this evening for Oxford Pint of Science's live event. Thank you to our wonderful speakers, so Anna, David, and then Charlotte. These events are not possible with a huge amount of team effort. So in the background, you may have caught a slight glimpse of her. We have Jess, who's been sharing all the screens. We have Lucy on the comments. Please keep your comments coming, and our speakers may wish to reply to you following the broadcast. And thank you for Nikki for hosting this event with me.
Thank you, Nav. Please stay tuned for more Pint of Science from the University of Bath in half an hour.
My name is Adam. I will be your quiz master for this evening. I certainly hope you're going to enjoy yourself. Here we go.